大家好啊，欢迎回到临床小白牙。今天呢，我想给大家开始上传我最近跟 CEO 的一些闲聊吧。然后呢，我给咱们这个系列呢叫做 See Your Pick。那希望呢，大家也能跟我一起来聊一聊、挑一挑，感受一下 C level 的人到底都是怎么想的。那第一期呢，我们来聊一聊 Sethera Therapeutic 的这个赛道的 CEO。那他们比较重要的是一个 platform 的 company， 里面会有一些故事是减肥药口服肽的赛道。那这是我的免责声明，这个呢是专门为这种访谈加的英文的声明，因为CEO 们都是讲的英文。那首先呢，我在Bio 上跟这个 CEO 聊的，然后他呢本人是犹他大学的化学博士毕业的，他2023年才毕业的，非常的年轻，就直接转化为科学家、创始人跟 CEO 了。我在剪辑他的采访的时候呢，觉得他非常的幽默，非常的 humble， 也有很多科学的热情，当然他的技术底层逻辑也是非常的好。那我个人呢还是很喜欢他的，所以我就先把它剪出来了。
I'm Karsten Eastman. I'm the CEO and co-founder of Sethera Therapeutics. Mm-hmm. We are a platform company that is working right now with peptides. Okay. We use enzymes to hyper-modify peptides to make them more drug-like at the outset of screening. Peptides right now are both a warming and a hot topic in biotech companies. Yeah. You have a lot of the commercial success with these GLP-1 therapeutics that have been fantastic for helping people with weight loss and diabetes. But one thing that people might not realize is that things like insulin are also a peptide. So a peptide is a small chain of amino acids that are built together that can have some very potent biological effects. Peptides are a little bit larger than small molecules, but they are quite a bit smaller than biologics or antibodies. So we're kind of in that gap, in that space in between small molecule therapeutics and the biologics. So we are utilizing enzymes to make peptides more rigid and more constrained so they can spend more time on target or be more specific to the target of interest.
那他们呢，其实在制药界呢，它解决的一个问题是什么？有超过 80% 的治病的靶点呢是不可成药的。原因呢，简单的可以理解成传统的小分子了，它够不着。那大分子的抗体呢，因为各种原因呢，它进不去那个空间。所以呢 Sethera 的解法呢
The way that it works is we will make massive libraries of linear peptides. Peptides normally are like linear chains, kind of floppy, they'll move around.
I see.
What we do is we will take that library of linear peptides and then use our enzyme. Our enzyme is a molecular machine to basically staple together the peptides into specific shapes and specific forms.
Will that peptide still work if you change the shape?
That's the really neat thing is a lot of peptides in nature are actually stapled or modified like that. So in your body, you have peptide hormones that have that same type of architecture. What we are doing is we're taking some of the lessons that we've learned from nature and some of the lessons that we've seen in really powerful bioactive peptides and then screening those same architectures in mass.
Oh, okay. So you are getting your artificial, if that is an artificial peptide, but learning it in a way that will be stabilized in nature, and then you make your peptide.
That's a good way to think about it. So yeah, we take a lot of the architectures that nature has shown to be exceptionally biologically active.
Okay.
And then what we do is we mimic those architectures, but then change the identities of the residues in between. So we have a big architectural breadth, and then also we have a very large chemical depth. Because we can change the identity of the side chains, and the side chains are what gives it a lot of its chemical identity.
那个 linker 的技术开发的一个高稳定性的、高结构多样性的多环大环肽英文也给大家写在这儿了。 那它的核心优势呢，它是具有大分子的高特异性跟小分子的强组织的渗透力和稳定性的特点。它是怎么做到的呢？因为呢，我们知道 peptide 啊，它是一段的蛋白质叫做 peptide， 然后呢，它很容易被体内的蛋白酶呢以为它是没有用的，因为它够小嘛，就迅速被降解了。比如说我们很红的这个 GLP-1， 它其实是一段 peptide。它这个减肥药啊，它为什么需要大量的反复的频繁的注射呢？就是因为打进去之后呢，它其实很大一部分呢是被迅速的降解掉了。那这个解法呢，就是它通过它这个 Sethera 的他们有专利的一个特殊的酶，它可以做到指哪打哪做个 linker， 像这个你看一个直条形的一个 peptide， 它可以做一个环形把它 link 起来，还可以 link 不同的地方，对吧？那这个时候呢，它就会变成一个比较复杂的稳定的环状的一个结构。大概的意思就是说，你越复杂的东西你就越难被分解。但是呢，你在把它卷起来的同时呢，你要把它的特异性以及它们的 functional 的这个 domain 啊，这个靶点呢，还是要 expose 出来。但是呢，本质上就是这么一个逻辑，就是你要卷，但是你要卷好，把它 fold 好，就是这么一个逻辑。
You were mentioning you are a platform company. So can you leverage us a little bit that do you manufacture for other company when they have the idea and then ask you to do that, or do you have any lead assets you may want to put forward?
So it's a little bit of both right now. So currently we are working towards developing 2 assets towards the clinic. One of those assets is going to be leveraging the congressionally directed medical research program funding that we were recommended for. So we're trying to do that. But then we have another asset that's going to be in the inflammation and immunology space. That's going to be around TNF and TNF pathways. But at the same time, we've been actively engaging with other large pharma in order to try to find what are the targets that they want screened, and then we can develop new hit and new lead molecules for them. So really the idea is advance internal assets, but also be a partner for big pharma that has specific targets that they'd like to hit.
然后它的核心赛道呢，它其实更专重于是一个新药的研发的技术平台，它是基于这个自由基的酶啊，多肽的交联的技术，具体我就不深入展析了，我也没有懂太多，只是很基础的东西。但是呢，本质上来讲，他们的平台呢，就是可以精准的安装这个键，使单个多肽呢，最多可以变成6个大环，而且呢，它们可以兼容一些非天然的氨基酸。这个呢，在他的访谈后面会有谈到的。因为这个重点在于什么呢？因为有一些自然存在的 peptide， 那我们把它改装 OK 没有问题。但是呢，它有些非自然存在的氨基酸，它是有一定的药用原理的。但是它为什么非自然呢？很多一部分就是它不稳定，它不可以存在太久，所以呢，自然天然的氨基酸没有它。但是呢，我们要想用它怎么办？所以呢，我们如果说这个平台能够使这些非天然的代谢不稳定很快的一些氨基酸能够 stabilize 的话，它的序列以及结构的空间组合就能够成指数级的扩展，大概是这个意思。那这个无限的想象力呢，是由这个极高的稳定性带来的。它改变了这些给药的方式，像绿色的这里有一些只能注射的多肽因为注射呢就直接进血液嘛，它就可以去到病灶很快。那如果说它够稳定的话，我们就可以口服啦，或者是超长效化。其次呢，是多靶点的协同。如果一个多环肽可以设计不同的环，同时能去咬住多个致命靶点的话，像我们之前聊过的 bispecific antibody，就是 SMMT 嘛，大概是那个意思，把2个目标的 细胞拉在一起，所以一个多环肽呢，设计不同的环，同时去咬住致命靶点呢，大概也是类似的这个意思。那所以说呢，它在application 里面呢，比如说举了一些例子是复杂的肿瘤学啊，针对一些检查点或小分子难以结合的蛋白作用的表面啊，或者是罕见病和多靶点的同时协作，那这些呢都构成了这个平台的无限商业的想象力。
Do you have any proof of concept story you can share with us?
Sure. One of the really neat things is we were able to show that our peptides are stable in simulated physiological conditions. So we took one of those peptides I mentioned that is cyclized by your body or in nature, and then it also took our same version of it that has our cyclization technology. After 30 minutes, this had completely linearized and was no longer active, and after 18 hours, our peptide remained stable. Now, this same peptide we tested against the receptor that is activated in the body. That peptide still activated the same receptor. The other piece that we've done is we've taken the libraries and screened them against 2 different oncology targets. And in those cases, we've pulled out single-digit nanomolar binders, so incredibly powerful binders. But what's neat is when we compare our peptides against other macrocyclic controls, macrocycles just, peptide that's a circle, our peptides stay on target 40 to 60 times longer. So in an oncology space, the reason why this is important is if you have a peptide that has a warhead attached to it, which is one of the ways that they treat types of cancers, you want that warhead and that peptide to stay attached to the cancer target, not to pop off. So our peptides are extremely good at staying on target.
Just follow that question. Usually, oncology people listen to that sentence, they will ask, like, how about the toxicity if your peptide stay there forever?
Well,
what's the mechanism to hop off?
So they will only stay there as long as the target is present. And if you've killed the cancer cell, then in that process, the target on the cancer cell is no longer there. So the peptide won't remain there.
I see. So will the peptide get metabolism or digest?
That's an area that we need to do additional research and continue to prove out the platform. What we've done so far is some really early proofs of concept showing that we can do the screens. The screens produce peptides that are active or hit the target, and then the targets that we've hit, the peptides stay stuck to them quite effectively. When we start moving into some more advanced areas like PK/PD, that's where we would need to do some additional work and additional research. This was just a cancer example, but we do have examples of some of our peptides that we've taken into mouse models now, and in that case, in diet-induced obesity mice, our peptides have at least shown to be very safe. Like they haven't caused any adverse reactions in these early animal models.
Great. So can I ask, in your stage, from my understanding, your proof of concept is all done in vitro, and you mentioned some of the study is in vivo already. Is your lead asset IND ready or
No, the lead asset we're still working on putting together our IND enabling package. That's where some of these early animal model data is still quite early. Yes, since we are a relatively new biotech company, one of the first points that we wanted to prove was the breadth of the biology that we can address, which is why we first wanted to get screens and confirmation in oncological spaces, immunology, and rheumatoid arthritis spaces, and then also the metabolic space and infectious disease.
Wow, very ambitious.
Yes, but that's what we've done already. So we have successful screening campaigns in all of those areas, and all of those areas have produced some very unique and interesting hits.
I see. So let's think back for just a moment. What can it do at the end? What's your dream? Can you talk about that?
So I think a really powerful example here is actually insulin. I mentioned earlier, insulin is a peptide. There's been a true tour de force, both on the academic side and the industry side, to try to make single-chain insulin. And there's been very limited stories of success. One of the biggest lessons in Biology 101 is structure equals function. And you have a lot of these peptide platforms who are limited to a macrocycle. And this is one structure, Sure, you can have all of the chemical diversity on the outside of this structure, but it can't address all of the complexities of biological surfaces. Because Sethera is positioning itself as an architecture-first type screen. It means that we can go after many protein-protein interactions or many complex surfaces that small molecules and simple macrocycles can't address. The other point of this is we are also specifically tailoring our peptides to be cell-penetrant. One of our big hypotheses has been it's not just the identity of the amino acid that enables, say, a penetrant peptide, but it's the architecture of the peptide as well. So what we've done is we've built many architectures and started to find the ones that enable peptides to get into cells, which enables entirely new classes of first-in-class and then also potentially best-in-class, because there's areas that small molecules can't address, of peptides. So we're working on orally bioavailable, cell-penetrating peptides that have complex architectures so they can address many previously undruggable targets.
So is there any topical interested as well?
So I think that there can be topical interest, especially in, certain types of cancers, right? Where you can have this really potent peptide that can penetrate the skin and can address just the cancer target in the skin. That certainly is an area that I view as being very amenable to our platform, but it's not an area that we've explored yet.
Just curious, your business stand now right now?
Right now we're more focused on actually a seed round. We've been
seed
we've been operating on very efficiently on capital that we received from an initial friends and family. But with that friends and family round, what I'm saying is we've been exceptionally successful with what we've done. We've screened 7 very diverse targets. We have peptides that are already in animal models, and we have great cellular data and cellular readouts. So really the next step is we're going to take 2 assets and start advancing them towards the clinic with the next round of funding.
So is that you want to licensing out to the company, or you just want a VC?
Well, that's again where we're actively pursuing potential big pharma partnerships. So we have a number of meetings set up where there's some interest in the technology, and I'm continuing to prove out the different points that big pharma has asked us to do, and I think that we have great data there supporting it. And then at the same time, I will recognize that if we do want to advance assets towards the clinic of our own, and that will take some additional funding.
Do you leverage any AI too?
No, and one of the really neat things about our platform is we are absolutely upstream of AI. And because the architectures and structures that we're generating have never existed before in nature. So there is no AI that's been trained or can even start to recognize what our structures are because they've never existed.
But that sounds very interesting though, because when I imagine entire process, you have a linear peptide and then you maybe like this way, and then your link will be here and there. That is a lot of data. And somehow you may want to establish your own database.
That is what we're planning. So in the next round, we actually have funding set aside that we will specifically use to build our own AI and ML based on our architectures. We have hundreds of terabytes of data supporting, both the peptide structure, the interactions with targets, and how those readouts look. So we're sitting currently on a wealth of data to start building our own AI and ML. There are many peptides right now that are actually quite expensive to produce. But then if we bring this back to my favorite example that we keep talking about, insulin, it's very cheap to produce, right? Because it's done recombinantly. So insulin is done in a link and cleave style. And you have to use an enzyme to cleave insulin, which is very analogous to our process. But rather than using an enzyme to cleave, we use an enzyme to build an additional architectural scaffold. So in the same vein as making a therapeutic as cheap as insulin is to produce, we are also starting to fund an effort to make insulin-like manufacturing economics for the peptides that come from our platform. Meaning that for a dose of peptide from our platform, it would cost, on the order of 5 to 15 cents, rather than the solid phase peptide support that is necessary and much more expensive.
I think you may need to educate me a little bit on this claim.
Sure.
So you have insulin is like, for example, $1, and how your cost can be cheaper using. I'm just imagining you need to have more material, like at least you need to add an enzyme and link. Will that be because it's more stable and temperaturized?
Insulin still uses an enzyme to reach its final active form, right?
Yes.
It has to be produced, and you have to use an enzyme, right? What I'm saying is, similarly to insulin, we can produce our peptides through recombinant means, and then use our enzyme to do the final tailoring step.
Oh, I see. So it's kind of like the CMC process.
Yes.
It's more simplified. Got it. Okay. But you will need to prove it's as safe, as clean as.
Of course, and that's one of the necessary steps. One of the things that we've been working on is actually a new enzyme immobilization strategy.
Wow.
So rather than needing to worry about the enzyme being a contaminant, the enzyme can all remain immobilized on a solid phase support, and it's just the peptide that will pass through. And because the peptide is a certain size, it's then extremely easy to filter a certain size peptide out, and nothing larger can pass through.
So if we talk to this stage, which direction you want to go? You want to have your resources to push your lead assets out, or you want to focus on your amazing platform and make it as a very useful platform to collaborate with whatever farmer?
That we did number 2 first. So this initial tranche of money that we were using, we focused solely on the platform and developing the platform to be as repeatable, robust, and unique as possible. And I think that we've successfully done that. The next step is really pushing an asset that came from the platform towards the clinic. That'll be a major proof point for Sethera, but it will also be a major proof point for the people that we want to partner with. Our goal here is to truly make successful medicines that will make an impact on people. And we recognize that there's a lot of expertise in these external companies that can help guide towards more successful medicines. Of course, we want it to be a successful commercial effort as well, but for us internally, we have certain expertise and certain key opinion leaders and certain advisors that we've brought on who have exceptional strengths. And we're leaning on those exceptional strengths first for our 2 initial programs, but then we also want to partner with other people who have a deep understanding of these other fields and can help guide and tailor these new and more successful medicines.
Okay. So talk about last little bit of for the audience.
Sure.
If they want to invest in your company.
Yes.
So the only way right now is via VC, like not an open market, right?
Right. So we're going to be opening up a new round. So you're right. There is not going to be an opportunity on the open market, but there will be an opportunity to start to have, say, a meeting with me. And if there's an interest in the company, be happy to, show our current investment plan and the use of proceeds and where we see the company, what milestones we would be able to hit. So I'm more than happy to take meetings with people who have an interest in peptides, peptide therapeutics, and an enabling platform technology that allows for multiple shots on goal.
Is there any plan for IPO, or it's just not in the scope right now?
So I think that that's an area that we would like to certainly move towards, but I recognize that where we're at as a company, we're a little early for that. But that's certainly something that I believe is a very achievable goal with what we've done so far. But I also recognize that I will need to do a lot of additional work in order to make that a reality.
Okay. Do you think the family funding you are operating with, do they want to still have a fully control of the company for?
No, so I think that was understood from the outset, is that this initial family funding is that they wouldn't have control. The control of the company has remained with the co-founders from the outset, and that we also recognize that if we move the company forward, there will have to be, some dilution of the current shares, because that comes with the territory.
Yes, got it. I think, yeah, sounds really good. I think it's really promising.
Thank you.
Of course, it's still very early stage.
It is. Yes, it is. Of course.
How do you feel right now as a CEO? Do you feel difficult to find partner, or do you feel, wow, people is all interested in my platform?
We have had a lot of interest in the platform. And I'm very happy to be able to say that. And the nice note is that with people that have had interest after the first meetings, they've still come back and been interested in additional meetings. Each time we've received different feedback that we've been able to use to further bolster the platform. But I think that we've made some fantastic success. We've screened really diverse targets. We have peptides in animal models. We have great cell data. And we're building on everything that we've learned in order to keep pushing it forward.
Okay. I'm going to dig into this a little bit because nowadays I think pet medicine is also a hot topic. It sounds like peptide can be easily have a branch on the pet medicine as well.
So like pets, like your dogs and cats. Yeah, and that's an area that we've talked to one partner on, and I think it's the same process can be applied whether it's a human target or it's an animal target.
Yeah.
So in terms,
especially if you build your AI platform.
Yes.
That might be something.
You have the human target, the animal target. You overlay them, can see what residues are different, so it's the same. Take the data from the human that we've already seen, can transpose it over to the pet side, and immediately have a potential good hit or even good lead candidate from that type of overlay. So I see your point. It's excellent.
I
from CMC background, so I'm just wondering. I know peptide is very one of the hottest area, and I'm sure you are doing such a great job. I'm just wondering, do you see your strengths over other competitors? Because I'm sure you are not the only platform working in the peptide library field.
So our competitors, and there are a number of peptide platforms, are all limited to macrocycles. So they have a single macrocycle. There's one competitor on the field or maybe more now, that's using constrained bicycles that look more like a heart, right? But here they can have one architecture, really. They try to play it out that they have more, but it really is just a macrocycle. So they can make this, they can have tons of chemical diversity, but Sethera is not limited to the macrocycle. We have a polymacrocyclic platform, which means that we can create all kinds of very interesting and unique motifs. And what we've done is we looked at, in biology, all of the interesting folds in, say, proteins, interesting folds from peptides in nature, and interesting folds that are already in therapeutics, and we built our libraries to mimic that. So we're not just locked into a macrocycle. We can chain macrocycles together. We can do nested macrocycles. We can do a macrocycle inside of a macrocycle, and all of this diverse architecture that directly relates to diverse already biological effect, and that's how we've built it, and that's how we're above everything.
举个例子叫 bicycle fiber product， 主要呢是化学合成双环肽，作用于 PDC， 就是 peptide drug conjugated， 就是用这个 peptide加上你的这个化疗的药物，把它运送到你的目标的地方，叫做多肽偶联药物。这个呢跟Sethera 呢，它不是直接对打这个平台，但是呢，它们技术有通用性。另外一个叫 PetDream， 它就用 mRNA 展示这个系统，是目前全球大环肽筛选的传统巨头之一。但是我稍微看了一下，他们的就是复杂度没有这个 Sethera 这么高。复杂度的意思是能把那个 peptide 卷成的结构的灵活度没有 Sethera 那么高。那它的差异性呢，我刚刚也大概讲了。还有一个在生产方面的差异性呢，就是它是纯酶生成的，因为它是纯生物的生成，它就避免了很多复杂的化学交联带来的副产物和纯化的难题，这个是纯粹在生产端了。 且它的多环架构呢，比单环跟双环更具有更强的空间立体防御力。我们刚刚讲的这个 bicycle 它是化学双环嘛，然后这个 Sethera 呢，它可以做到6个环，所以它的抗降解跟多靶点的装载力呢，它跟不是同一个数量级的。
One more add-on.
Please.
So is that like a simulations, like a stage for all the like peptides you are just described, or is that you are very thinking about like a CMC? How shall we synthesize all the molecules in the real world?
So we've already synthesized all of those
All of
molecules.
them? Wow.
So we truly are doing functional screens of hundreds of trillions of peptides in real space and real time. And those peptides all have unique architectures, but we have complete control over the architecture that they have.
That's impressive.
Thank you. Yes, we've done a lot but this is all enabled by enzymes. Standard chemistry processes would not allow for these types of architectures to be made. The only way these architectures can be made and screened is through the use of enzymes.
So you own the IP of the enzyme?
Yes, we do.
Great. Okay, thank you so much.
No, thank you so much. I really appreciate
OK， 那感谢你看到这里了啊，这是我第一个的 CEO 访谈的视频。然后呢，希望大家多多点赞转发，把我们这个频道的流量推上去，那我就有更多的抓手跟靶点，可以去访问更多的 C-level 的朋友。我也会继续努力把这个事情推进的。那就先讲到这里。如果你对这方面的视频，或者是你觉得这个流程上啊，或者是你觉得我问的问题还不是太得劲儿啊，欢迎大家在下面评论区，我会慢慢的把这个系列做起来的。